Okay, but the aim of this is going to be talking about compositional representations for flexible behavior. So we're going to try to understand a little bit about what the underlying computations are you might need for certain specific types of flexible behavior. We'll try to go from those computations down somehow to understand a little bit about neural representations as well. So the first thing I'm going to try to convince you of is that flexible behavior requires models of the world. You won't need much convincing, but I'll do it anyway. So here is, unfortunately, you can't see my mouse, which is a bit of a pain, but I'll do lots of pointing. So here is a mouse, and it's been trained to run around this barrier to receive this reward. And it can do that. It can do that. You can train it. It can just run around over and over and over again, and it will get some reward. That's great. However, if you open a new shortcut, then repeating those old actions are no longer the right thing to do, or the optimal thing to do. Instead, going through that shortcut is. However, this animal can only know that shortcut is a thing it can do if it knew that this location connects with that location. Okay? So in order to know that it can do that, it needs to have some underlying model of the world, how locations in the world connect with each other. Of course, you don't just want this for a single environment like we've got there, but this animal wants to be able to perform as optimally as possible in a whole variety of environments. For example, this particular configuration of walls and rewards. You want to be able to know when it gets plopped up there that it can run straight to the reward, and it wants to be able to do that as fast as possible with as little extra learning as possible. So what it wants to be able to do is how can this animal build a model of any world it enters as fast as possible? Because if it can do that, then it can reach any reward from any location and can solve all the problems. So one particularly attractive way of approaching this idea of how do I build a model as fast as possible is if I break down my understanding of the world into constituent building blocks. So for example, we've got the building blocks of space. Both of those environments have something about space in them. The building blocks of reward, both of those environments have reward in them. And building blocks of walls, both environments have walls. And just like how children, or adults if they want to, but just how children can take blocks, building blocks, and build them into one tower, they can put them in a different configuration and build them into different towers. And so that's what we want to figure out how to do. How can we represent our world in constituent building blocks and recompose them into a new configuration to understand any new world? So that's going to be the aim of the game. How do you do that? Well, or in particular, like, how do we represent these building blocks? And that's the problem we're going to try to face, and we don't know how to do it. We're lucky, however, that there are some neural evidence that tells us a little bit about how the brain might be doing that, or indeed it tells us a little bit about how the brain is doing that. So what you're seeing here is a top-down view of a box, and that black trajectory, squiggle, 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 is everywhere a rat has run around. And every time you see a red dot, that is where this one particular cell has fired. And this is a famous sort of cell. It's called a grid cell. And these cells, they really only care about space. They do nothing else. They just code for where you are in space. That's the thing. They are building blocks of space. There are lots of cells like that, building blocks of space. There are also lots of cells that code about objects. So this is a cell here that always fires whenever you're 5 steps to the west of an object, these white objects. And here's a cell that always fires when you're 4 steps northwest of an object. These are cells that only care about objects. So they care about every single one of these white objects. They only care about objects, don't care about anything else. Object-backed cells, building blocks of objects. So these are all in entorhinal cortex. And you also find border cells. They care about borders. They don't care about anything else. Building blocks of borders. The brain is representing this spatial world in terms of building blocks. Of course, that makes sense, because any world is a different configuration of these objects, these walls, etc. I've already lied to you. Sorry, I should say that. Apologies for so early on in this talk. But what I just told you about everything that's so beautifully represented in building blocks is not always true. Sometimes these building block representations, they sort of blend together. They merge together. So here's an example of that. On the left-hand side is what when the animal first entered that environment, down here, it was a grid cell. It had these nice sort of hexagonal-like fields, or triangular lattice-like fields. And this is an environment, so those 3 black dots are rewards. But after a while of this animal being in this environment, we can see that the fields of this cell, they change. The receptor fields, they change. In fact, they walk towards the rewards. This firing fields, they walk towards the rewards. And this is true not just in one cell, but in fact many cells. So here again, this firing field over there has walked towards the reward. So this cell is no longer just representing space like it was before. It's representing space and rewards together. So it's not like a building block of space anymore. It's just some weird building block of space and objects together. Okay, it seems to be representing what you and I think of two things rather than just one thing.
So James, does it also mean we can't trust it? Because tomorrow it's going to be something else, right?
Well, we can't trust that. So I hopefully show that we can trust something about it. Okay. Okay. It's well, yes. It changes science as much as you and I do. Okay. So that phenomena of cells sometimes caring about one thing and cells sometimes care about many things is not just unique to the entorhinal cortex, the hippocampal formation, what we just saw. It's also found in parietal cortex. So here is a cell at the top, and it only is active during this one task called the T-maze task. And it is not active during this SW, this steering wheel task. Okay, so this cell only cares about that. Here's another cell that only cares about the steering wheel task, doesn't care about the T-maze task. And this is true across the whole population. These cells either care about one task or the other task. And there's a few that seem to care about most, but most cells care about one task or the other task. Cells care about one thing at a time. We also find that in prefrontal cortex. However, that story is, of course, as we all know, not the full picture. We find mixed selectivity in prefrontal cortex as well. And we find mixed selectivity all over the brain as well. Cells sometimes care about many things, but they sometimes care about one thing. There's a real puzzle going on there. Okay. That puzzle is also a big thing in machine learning, and it's called disentanglement. That idea is called disentanglement, when we have Arthur here, who knows all about this. Okay, and so the belief of these machine learners is that there is we have some observed data, which is some function of some latent factors, where those latent factors themselves are independent. Of course, the aim of the machine learners is to take the data, which is all that they see, and infer what those latent factors are. They want to be able to, they like it when their latent factors are single neurons care about single factors, because then it's quite easy to interpret. This neuron is a neuron that cares about hair color, for example. Easy to understand. Okay, so what they tend to do is they have vector representation. So they take a vector representation of the input, they project it to some latent representation, and then they apply all sorts of constraints onto that latent representation. All sorts of constraints which say, I want you to be disentangled. I want this neuron to only care about one thing. Okay, all sorts of constraints to do that. And when you do it, it really works. So here's a neuron trained on some data of various objects. Here's a neuron. That neuron only cares about the Y location of that object. Here's another neuron that only cares about the X location. Here's another neuron that only cares about the size of the object. It really do work. These models, they do work. They also work in an interesting way, or somehow an analogous way, apparently, to infratemporal cortex. So now if I train exactly one of these models, these machine learning models, I train on lots of faces. We don't know what the true underlying factors of the face are, I should say, but we can train a model on these faces, and the model will produce some, when this neuron is active, it's saying, I believe that there's one factor, this factor is going on in this image. But interestingly, we come and present those exact same images to monkeys, and we record from infratemporal cortex. It turns out that single neurons in infratemporal cortex, they are well explained by single neurons in these disentangled models, implying that infratemporal cortex, even though we don't know what the true underlying factors of images are, implying that the infratemporal cortex is representing single neurons caring about single of these factors, okay? Because they align with the model, which is apparently a disentanglement model. Okay, something interesting going on there. Okay, so this is bringing us to a few questions. What? Back, okay. Oh, sorry. This is a problem with animations. Yeah.
I mean, if you showed like 1 million faces, how narrowly tuned? These neurons can't be narrow.
I didn't say that they're tuned to a single face. I'm saying that each face there can be expressed some by some latent representation. So it might be the case that latent representation has 10 factors, and each factor will be at some particular, some particular value.
I thought you said each face had a.
No, each face does not have a neuron. Okay, it's just that each neuron corresponds to some underlying generative factor of the faces, and therefore each neuron in the deep net does, and each neuron in the infratemporal cortex corresponds to some underlying factor of the face. It doesn't have to be a single face. So it could be hair color, it could be nose size, it could be etc.
Oh, okay. So that's. So So how can you tell that from this figure, or is that?
So you can tell it by this figure by we can look at the amount of variance. This is a one cell, sorry. The amount of variance that gets explained by one cell. The amount of variance that gets explained by single neurons in this.
That's a factor in
so this is so 1 to 10 is the number of neurons in this deep net, and this is how much that neuron explains about the cell. And so often you get a single peak, implying that a single factor found by this deep net is able to explain a single cell, and therefore there's some alignment between disentangled models and infratemporal vortex. Let's not dwell on it too much, though. Okay, but this whole sort of background I've given you raises some set of puzzles. Okay, the first one is why sometimes do these single neurons care about single variables, like these cells that just care about grid cells, or the cells that just care about objects, sorry. And why do we sometimes not? Why do we sometimes get this entangled situation like before? Is there some sort of general principle that isn't just for all this spatial stuff here that could explain those parietal cells, could also explain what those machines are doing? Is there some general principle going on, not just for space, not just for brains? And lastly, if we're interested in understanding these representations, why does a cell look like the way it does? Why does a grid cell look like a grid cell in the first place? Okay, and so we're going to try to answer these questions in this talk. And I'll run through two pieces of work on this. Okay, and the way in which we're going to think about this is in a very similar way to what these machine learners were doing. We're going to think about what are the constraints on neural representations. We're going to formalize those constraints and see where that takes us to, in terms of what the representation should look like. Okay, so what are some constraints on neurons? Well, firstly, neurons have to be useful in some way. Okay, so if we have a task which is in some way composed out of X, Y, and Z, or Z for the British people, then if we have a neural representation which is somehow encoding that task, that representation, so these are neurons, meant to be neurons, these colors meant to be activities, but these neurons should somehow know about X, Y, and Z as well. Okay, they need to contain some information about X, Y, and Z if they're solving the task. Neurons need to be useful. Second set of constraints that we're going to consider here are constraints what biology imposes on a representation. Okay, so biology says that neural activity cannot be negative. So that means we're not allowed something that looks like this. Neurons can't be spiking negative, but we are allowed something that looks like this. It's also the case that the brain has finite energy budgets. Okay? So we don't want to be spiking like crazy or high activity. We want low activity. We don't want our synapses to be huge, or synaptic weights to be huge, because that costs energy to do that. Okay, so these are constraints. There are of course more constraints going on, but these are constraints we're going to consider here. Useful, obey biology. I'm going to now try to describe to you that these, or offer some intuition that these constraints lead to disentangled representations now. Okay. So what I'm showing here is that we have two neurons, and these two neurons are trying to represent this distribution. Okay? So they're trying to represent this distribution, which is described by two independent random variables, E1 and E2. They're representing it in some nice, simple, linear way. And what I'm showing here, I should say quickly, so we can see that E1 is projected along this axis, and E2 is projected along this axis. That's described by the equation at the top. And over here, we have the energy, the sort of amount of activity, the amount of energy that these neurons are using to code for these two random variables. Okay, so they're managing to code for the random variables, so that's all very good. But of course, the problem is that they both have negative activity, and that's not allowed by biology. Okay, so what we can do is we can shift this whole distribution up. And that's great now. We've got no negative activity, but it's come at an energy cost. It's come at an energy cost because these neurons are now they're firing faster. They're more active, more energy. Okay, so maybe it's clear that the way in which we can reduce this energy burden is that if we take that gray square and we squish it and we stick it into the axis as close as possible. So we do this. And that indeed is actually the best way that you can do this. But something interesting has happened when we did that. Something interesting is now these random variables, they're projected along neural axis. So E1 is projected along this axis. E2 predicting along this axis. Single neurons care about single factors of variation. Intuition makes sense? Okay, great. I'm always embarrassed for this, but okay. We can also, to be an arse, we can also write this all up in maths and prove it. Okay.
So,
at least for the linear settings.
This
yeah.
requires bounded distributions.
There are some constraints, yes. So one of them exactly is that distributions have finite support. Yeah. And this is one
lives in a circle. Does it
lives in a circle. Well, that's no, that won't work. Yeah, I don't think. Yeah, okay. But I don't think. Yeah. Circles not independent. So they need to have independent variables. So linear transformation independent variables. Circles not independent.
Gaussian has been Gaussian.
Gaussian's not allowed. It doesn't have finite support. Yeah. Okay. So that was sort of a theory, or at least I showed you some intuition behind why biological constraints might lead to single neurons caring about single factors of variation. Does it actually work in practice? Okay, so we're going to train some neural networks and with these constraints and see if those neural networks then inherit these properties of single neurons caring about single things. Okay, so we're going to take some input and output data. So input data, we're going to have some X data and some Y data. X and Y are really scrambles of these independent variables. And I try to visualize that by each neuron has multiple colors, so each neuron cares about multiple things. But there really is some true mapping between X and Y. And we're now going to take a one-layer neural network, and we're going to ask it to take in Y, take in X, predict Y. You can see that X and Y, and they're both mixed at the moment. But critically, when we train this neural network, we're going to ask this hidden representation here, we're going to ask it to be non-negative, okay? We can do that in a variety of ways. We can put like a ReLU activation function. We can add a regularization cost saying don't be negative, but we can do it in a variety of ways. We're also going to ask for the activity of the hidden layer to be small. Don't fire fast. That's one of our biological constraints. And another biological constraint of energy efficiency, we're going to ask its weights not to be too big as well. So train the network, biological constraints of non-negativity, energy efficiency. Okay, so, and we're going to ask what happens to that middle layer. Is it mixed or is it disentangled? Well, if we didn't have these biological constraints, it learns that function just fine, no problem. But we can see from here that neurons care about many things. So this is a matrix where these are neurons and these are the true underlying factors. Okay, so we can see that this neuron, for example, cares about this one a lot, this one a bit less, this one a bit less. This neuron cares about that one, that one, that one. Neurons are mixed, they care about lots of things. However, when we have these biological constraints, single neurons care about single things. This neuron here only cares about that one. This neuron only cares about that one. Disentangled representation. Single neurons care about single things. So that's just like a toy data set. And I guess I'm going to show you another toy data set, but just slightly less toy. And we're going to test this on a data set which is a machine learning benchmark for disentanglement. So this data set consists of lots of images where each image has really been generated from one of six, well, not one of six, as a combination of these six factors. So each image shows an object where that object is a certain size, a certain shape, a certain color. You're viewing at it from a certain angle, and the wall and floor colors are different. We have lots and lots and lots of these images. And then we're going to come along and we're going to train one of these deep net models where we take in some inputs, predict some latent space. But now we're not going to use all these funky constraints that machine learners have. We're just going to put our biological constraints on there as well. So we're going to ask it to be that representation to be non-negative and have energy efficiency.
Is this supervised or unsupervised?
This is unsupervised. So yeah, it's a sort of variant of an autoencoder of anyone. Okay, so we're going to do that and we're going to see what happens. And I'm not going to show any numbers, but I'm just going to show a few pictures. Okay, and so we can come along and we can visualize what single neurons care about after we train one of our models. Let's look at the first neuron. The first neuron, it just cares about viewing angle. So as you scan through that neuron's values, all it cares about is viewing angle. It is only representing viewing angle. Second neuron is only representing object shape. It's going from sphere to cylinder to a cube. Then the third neuron only cares about wall color. These neurons don't care about anything. Floor color, nothing. Object color. object size. Okay? Each neuron only cares about one thing. That's a disentangled representation. It also turns out that, I mean, this is probably like a year out of date now. What is it? Is out of date. But it turns out if you compare these models where you have our biological constraints to one of the popular models in machine learning, I'm not going to explain everything that's going on, but essentially all of the ones with biological constraints, they fall into this Goldilocks regime where they both disentangle really well, quite well, and they also account for the data quite well. Okay, so sort of Goldilocks period. Whereas these machine learning models, they often account for the data, don't disentangle, or they disentangle and they don't account for the data very well. Okay, so some Goldilocks zone. I'm not claiming this like it's going to be the greatest thing in machine learning, but it's an interesting phenomenon. Okay, so that was all simulations. What about brain representations? Yes.
Do you have any measure of energy difference between the biological constraint, non-biological constraint?
I'm not on hand, but there will definitely be lower. Yeah. But I don't have that on hand.
See, like there is a very big difference for the loss.
Yeah, I don't know. So like the next current best model from machine learners, it's actually directly inspired by this, does use like huge energy regularization now, and that does do very well. But I don't have like pictures to show the difference. Okay, so that was all models and theory. What about brain representations? Which is also going to be some models and theory, but we'll relate it to brains. Okay, so I'm going to try to show you exactly the same principles that we just saw. Can also explain why cells sometimes seem to code for these building blocks and why they sometimes blend together. Okie dokie. So just like these machine learners, they had a data set which was composed of those 6 independent factors for those images. We're going to make a data set now where space and objects are independent from each other. Okay, what does that mean? So sometimes we can have an animal, it's going to run through an environment like this, run. And in that environment, it's running in the spatial environment, but there's some purple objects in there. And sometimes it's going to run through another environment which just has a different spatial arrangement of objects. But critically, it's going to run through lots of these environments. Every environment is going to have a random arrangement of objects. Space and objects are independent from each other. And then we're going to come along and we're going to train our current neural network. We're going to ask it to predict where you're in space. Is there an object there? So this network better learn about space and objects because that's what it has to do. And we're going to see what happens. Well, without these biological constraints, we find cells, so these white dots are hard to see, but they're the objects. We find cells, these are 2 cells, and they seem to look a bit grid-like, some amorphous grid-like. And this is without biological constraints in a task with objects. In a task without objects, those neurons sort of look kind of similar. I promise you they're actually not exactly the same, but they sort of look similar. Somehow they're coding for objects. Not obvious how. However, with biological constraints, these are a task with objects. I'm showing 4 cells now. We either find cells that look like grid cells, or we find cells who have this sort of vector representation towards objects. They're like object vector cells.
Sorry, which of those on the right is without objects?
So we haven't looked at that yet. Okay. Currently, we're all in. These are 4 cells from a task with 5 objects. Okay. Either find things that look like grid cells or things that look like object vector cells. In tasks without objects, grid cells are identical, no change, because they only care about space. They couldn't care less whether they're objects or not. But the things which cared about objects before turn off. Okay, they turn off. There's no objects there. There's nothing to care about. Okay, relates to grid cells or object vector cells from before. Okay, so we've seen that when you have underlying task factors that are independent and these biological constraints, there is modularity in our representation, disentangled representation.
Yeah, does that mean it's question?
It does, but I've chosen to temporarily ignore it because okay. I might ask.
No, I'm not.
Okay, well I've entered into a slight pickle now because I'll move that over there. I can't see the mouse on my screen, so I'm sort of I don't know where the X is. Okay. Anyway, so what about that warping phenomenon that we saw? Okay, so oh sorry, okay, I've spoiled it now. Okay, but now we're going to train on a task where there's only one configuration of space and objects. Okay, we're not going to have all of those tasks. We're just going to only train on this one task with 2 objects in it. Do the same thing, and then we can look at the representations we learn in this model. And we find cells that kind of look quite grid-like. There's something sort of funky going on. And let's look at this example cell. We can see that it's always got sort of the same vector around an object. The firing field's always sort of got the same vector relationship to that field. This one's always, the firing field's always to the southeast. but it's not an object vector cell because there's also these other firing fields going on like that. So some sort of weird mix between grid cells and object vector cells. But all of the cells are like that. They all have a sort of preference towards an object, but then they also have a grid-like property as well. Okay? That is this warping phenomena that we saw earlier. It's exactly like these cells. They were grid cells, but now they're sort of like these weird warped grid cells. Okay? I've tried to explain. Sorry, no, I can't get rid of this thing. So apologies. No, I know if he's okay. Okay. But one thing I didn't tell you about this task is that the animals got put in here and then the cell at the very beginning in that environment, and then they have that cell recorded at the very beginning. Then after a while, this is after like quite a long time of the animal being in the environment, and all it does is just it runs around these walls, runs around the wall, runs on the wall. So to that animal, space and rewards have become entangled because all it does is just does the same thing in the same environment over and over and over again. So it's perhaps an explanation to why you get that warping. Okay, so let's revisit these questions. And first question was, why do sometimes single neurons first care about single variables? Well, we saw that hopefully biological constraints and biological constraints plus a sort of task with independent factors leads to neural modules. Why sometimes not? Well, we saw that we had an entangled task with this warping. Is there some sort of general principle not for space and not just for space and not just for brains? Well, we have that theory, which apparently works in these machine learning tasks. Why does each cell look the way it does? Well, we haven't answered that at all. Okay, so that's what we're going to try to have a look at now. Okay. I might just put a slight effort to remove this thing quickly. Okay, so in order to understand why grid cells look like grid cells, or explain something about grid cells, I have one thing I need to tell you, and is that these grid cells come in discrete modules. Okay, so we don't just see one grid cell with that sort of characteristic spacing between the peaks. We see multiple characteristic spacing. So this one's got these sort of low spatial frequency, high spatial frequency. And for each one of these spatial frequencies, which really is a discrete spatial frequency, there's lots and lots and lots of cells that have the same frequency. Okay? So this thing would be called a module of cells corresponding to a particular spatial frequency. Okay, so the question we're trying to wonder is why discrete modules? And to do that, we're going to need to formalize 2D space. So that's what we're going to do in the last 10 minutes or so. Okay, so formalizing 2D space. And, well, face is covered. This is work with a PhD student already mentioned today, Will Dorrell from the Gatsby. Okay, so formalizing 2D space. Right, well, we need to formalize it by what are the underlying rules of 2D space. So I guess the underlying rules of 2D space is like if you go north and east and south and west, you're back in the same place. And that's, of course, true whether you started at that location or you start here or you start here. Okay? Those rules are true wherever you're in space. Those are the rules of space. And therefore, if we have a neural representation that is coding for a location, then when we go east, south, west, and north, we want that neural representation to be the same when it's back in the same place. So we want our neural representation to track somehow where we are in space. This is a property known as equivariance. And we're going to formalize these rules. I'm not going to go into any nitty-gritty detail. We're going to formalize these rules with group theory and representation theory. What I am going to show is some equations. But I'm really just going to try to offer intuition at every stage. Okay. I'm not going to go into any proper details. Okay, so what this theory says, if I have some representation, some neural representation or vector representation of where I am, that's location x, then in order to get to a vector location of x plus delta x, there is some matrix, which is a function of delta x, okay? What this group representation theory says, it tells us exactly what that matrix is, or it tells us some properties about exactly what that matrix is, okay? And we're just going to use the fact that we have some theories, very well-developed theory, which tells us about what the matrix looks like. Of course, we aren't actually interested in matrices. We're interested in representations. We're interested in what the neurons look like. But of course, when you know what a matrix looks like, and you have a simple relationship here between where I am in space now, where I am in space now, we can get some, that understanding when a matrix constrains the neural representation of space as well. And it looks like this following form. Okay. You can look at that and think, oh, that's just a Fourier series. Like, what's he saying? That's nothing. Okay. But there's a slight nuance. Okay. It looks like a Fourier series, apart from we're capping at D. Okay. We're only allowed a finite number of frequencies. And the reason we're only allowed a finite number of frequencies is because we've got a finite number of neurons, okay? And so each frequency that you choose to represent takes up 2 neurons. There's a reason for that, but I won't go into it. Well, I could go in, but it takes up 2 sentences, which I've just taken up by saying this. Okay, okay, Fourier series, but we're capping the number of frequencies. And of course, the question is, right, well, this still could be pretty arbitrary. The question is, what on earth do these values of A and B take? If we know what those values A and B should take, then it will tell us exactly what our representation should look like. Okay, I'm going to do that. We're going to look at these constraints again. So a reminder of the constraints. So first one is we need to be functional. Okay, so we need to represent location. And we've worked out an equation that does that from group and representation theory. But we also want to do it precisely. You don't want to represent location very weakly. You want to be quite precise. And I'll get back to that a bit later on. But the first thing we can note is that if we were trying to represent location, then A0, that's a constant across space, so it doesn't help us represent space in any way. It doesn't actually discriminate 2 points from each other. So A0 does not help. Second point, we want to obey biology in some way. Okay, so the same one, each neuron can't be non-negative, same thing as before. And we also want activity energy to be small. So we want our neurons not to be firing too much. But now we've got an equation at the top. We can actually just solve for the activity energy. And this is what the activity energy looks like. Okay, and of course this will make sense. It's just the sum of the coefficients. Okay? But now we know something. We know something. We can work something out already, and that is A0 doesn't help co-location, but it does take up energy over there. So we want to make it as small as possible. We want A0 to be as small as possible. That's something we know already. Okay. Okay, those are things we know, but we're now going to think about how non-negativity might interact with all of this as well. Again, just going to try and offer some intuition. Okay, we know we've got this functional form, plus we know we want A0 to be as small as possible. And we're going to try to understand how non-negativity interacts with this by considering a single neuron, which currently is to be one element of this vector. And we're just, at this moment, we're just going to give this one element of the vector a single frequency. It's representing a single frequency. Of course, that single frequency is not negative, so it's not non-negative. So one thing we could do is we could add a constant value to it and shift it all up to be non-negative. But that takes up energy. So if possible, we want that neuron to we want to be able to add. If we can make it non-negative by adding different sines and cosines, that's preferable to us because sines and cosines still are useful for discriminating locations. Okay, so what we can do is we can add a single frequency to that. Of course, the frequency that we want to add is the one which has peaks at the troughs and doesn't knock down our peaks. And that frequency is just twice its original frequency. So we add on this orange curve twice the original frequency. And once we do that, it really is the case. This is slightly less negative than before. So we've done well. And we can keep on repeating that process over and over and over again, adding another harmonic, and so on and so on and so on. Until at the very end, you can have very, very many harmonics. And in the limit, it really is the case that you can get A0 down to 0 by adding as many harmonics as possible. But we have a limited number of frequencies, because limited number of neurons, so we can't do it forever. But nevertheless, if we look at the characteristic sort of spectrum drop-off of the amount of frequency content we want, it sort of falls off in this sort of linear way. Okay, well, we can come along and we can now simulate, this is what some theory is saying, we can come along and simulate some neurons. And indeed, if we simulate neurons, it really does have these like multi-peak things. It looks a little bit like that. We look at the frequency content of that, it really falls off. Okay? It really looks like this, our explanation here is what simulation finds as well. So it seems like theory is matching simulation. Okay, something interesting's happened, which I'll try to explain as well. In order to make this cell non-negative, we needed lots of frequencies. We needed lots of frequencies, and those frequencies are all harmonics of each other. But if you remember, I said that we're limited by the number of frequencies we have in the population. So if we want every cell to be non-negative, that means every cell needs to have some set of harmonics, but we're limited by the total number of frequencies we can have. Therefore, every cell will want to reuse the same harmonic frequencies together. So you can spread those frequencies across all of the cells, you can reuse them, and you can make them all non-negative. Okay, but now all the cells have got the same harmonics, therefore they're all going to look roughly the same, but they're just going to be shifted copies of each other. That is a single module of grid cells, a single module of the cell, I should say. Indeed, when we simulate lots of cells, They all look exactly the same, and they all have that sort of same characteristic frequency drop-off. They just have different phases. Okay, this same argument works exactly for 2D. Spectrum in 1D here, spectrum in 2D it will just drop off in 2D K space. But of course, many lattices of 2D lattices are allowed. This lattice is allowed, hexagonal lattice is allowed, some sort of other shared lattice is allowed as well. But the one we're going to take, a bit of a weak argument, I admit, is that the hexagonal lattice is the best. Hexagonal lattice is the best one, so it fits in the most frequencies. Fits in the most frequencies, more frequencies, discriminate points better. Okay. And indeed, when we simulate that, we get these sort of grid-like fields as well. Okay, so that's the single module. But why do we see many modules? Okay, so remember I said to be functional, you need to represent location, you need to do it precisely, but we'll get back to it. We're getting back to it right now. And we can think about what frequencies, we already thought about what frequencies that a single neuron wants. It wants harmonics. But what frequencies does the population wants? Well, if it had harmonically related frequencies, these are two frequencies in the population. It means that they're not distinguishing these 2 points. It's representing the same point, those 2 points indistinguishably. And therefore, we're not, we don't know that, this location is different from that location. Harmonic frequencies are bad for representing locations. If you had non-harmonically ones, every location is represented by a different combination of these frequencies. Therefore, we're able to discern points in space better. Okay, non-harmonic, yeah, exactly. Okay, so there's some interesting battle that's going on. So population wants these non-harmonic frequencies because it can decode points better. But single cells, they want harmonically related frequencies so they can be non-negative. That's a battle. And the way you can solve that battle is by modules, okay? So you have these modules where each modular cell, each module has lots of the same frequencies, so you can be non-negative. But across the population, you have lots of non-harmonically related modules, so you can still decode location better. And some evidence towards that, we can train lots of models. We can trade off this parameter, which essentially trades off how important these two terms are. We can see that when one regime of the parameter, you get lots of modules. Another regime of the parameter, you only get one module. Okay, so summary, we sort of axiomatize neural representations by this constraint view of things. We use it to explain grid cells. Also applies to lots of other variables too, anything you can represent with a group. And that's it. And thanks to everyone, in particular, Will, I spoke about before.